the catalase test is one of the first tests used when you identify gram-positive cocci. Once you know that a certain species is gram-positive cocci, you next need to do the catalase test. Why do we do the catalase test? After seeing the gram-positive cocci, the next question is it Staphylococcus or Streptococcus? And catalase test basically answers this. What's catalase? Catalase is an enzyme that breaks down toxic hydrogen peroxide. I'll show you the catalyst test here. So once the catalyst test is done, if it's negative, you know it's Streptococcus, and if it's positive, you know it's Staphylococcus. The Streptococcus basically exists in pairs or chains, and the Staphylococcus exists in clusters. Let me show this to you. All right, so what's the reaction? Basically, you take the bacterial colony and you place it on a slide, right? You add hydrogen peroxide to the slide and you watch for bubbles. So what happens is in a positive catalyst test, lots of bubbles appear. The hydrogen peroxide is released and oxygen is also released. So this means there's a lot of bubbles over there, right? So when you get bubbles, you basically see that this is a Staphylococcus species. Staphylococcus, I told you, clusters में appear करते हैं. They appear in clusters. So examples are Staph aureus, Staph epidermidis, and Staphylococcus saprophyticus. Now, a negative catalyst test will tell you that this is Streptococcus. This is not Staphylococcus. Now, when something is negative catalyst, you know this is either Streptococcus or Enterococcus. So you'll either have it as Streptococcus pneumonia or Streptococcus pyogenes or Enterococcus faecalis. We'll see it later on. But once something is catalase negative, you know that this is either the pairs or chain strep, or you know that this is either the clusters based staph. So this is a picture of the clusters of this staph, and this is a picture of the pairs or chains of strep, right? So when we went to strep, we said that it either be the streptococcus or it will be the enterococcus, right? And when we went to the staph, we said it will either be aureus, epidermidis, or saprophyticus. Now, in order to study the streptococcus species one level further, we need to understand the hemolysis test. Okay, so once the catalase test is done and you found out that it's your species is basically catalase negative, you know you're dealing with Streptococcus or Enterococcus, the next question is going to be what kind of hemolysis does it produce on blood agar? Now this is how we separate the different species of Streptococci. How does the species behave when we put it on a blood agar? Now, hemolysis is basically the destruction of RBCs, right? So the blood agar, our dish basically contains sheep red blood cells, right? So when bacteria grow on the plate, we look at what happens to the surrounding red blood cells because we have the sheep red blood cells on this agar. Now we have three types of hemolysis that the streptococci does. We have alpha hemolysis, we have beta hemolysis, and then we have gamma hemolysis, right? Now, um, let me just show it to you on this slide. So this is alpha hemolysis, this is beta hemolysis, and this is gamma hemolysis. What's alpha hemolysis? Alpha hemolysis means there's partial hemolysis. The bacteria partially destroys the RBCs, and the hemoglobin basically gets oxidized into methemoglobin. And this methemoglobin basically gives you this green color around the colony, right? So alpha hemolysis is basically this partial hemolysis where the bacteria partially destroys the red blood cells around it, and this hemoglobin gets oxidized into methemoglobin, and this gives the green color around the colony, right? So you have this green halo around the colony. Organisms that have partial hemolysis are basically Strep pneumoniae and Viridin streptococci. Viridin streptococci includes the S mutans and S sanguinis. We'll come back to that later on, right? Now, why is it green? I'll tell you because normal hemoglobin is basically Fe2 plus, and oxidized hemoglobin is Fe3 plus. So the Fe3 plus is called methemoglobin, and this produces the green color, right? Um, then we move on to beta hemolysis. Now, this is our beta hemolysis. Beta hemolysis is that the bacteria completely destroy the red blood cells. There is no hemoglobin remain around the bacteria. And there's a clear transparent zone in beta hemolysis, right? It looks like someone punched a hole in the blood agar, right? That is something I would want you to know. So, beta hemolysis is basically the bacteria completely destroying the red blood cells. There's no hemoglobin that is remaining, and it looks like someone punched a hole in the blood agar. Organisms that are beta hemolytic are basically group A and group B streptococcus, right? And that basically includes your strep pyogenes and your Streptococcus agalexia, right? Strep pyogenes is group A strep and Strep agalexia is group B strep. Now, beta hemolytic algorithm goes something like, okay, so I'll get back to the algorithms later on. By algorithm, I mean once beta hemolysis is done, what further tests we do to separate the pyogenes versus the agalexia. But I'll come to that when we do that later on. Okay. Then we move on to basically the gamma hemolysis. Gamma hemolysis basically means that there is no hemolysis. There is no color change and there is no clearing also. This is basically, we have two streptococcuses that do not change their color, complete hemolysis. This is basically seen by S. pyogenes and S. agalexii. But we'll look at it later on also, okay? Some Enterococcus species also do this. So this was your gamma hemolysis. This means no hemolysis, it grows in bile. And basically the non-Enterococcus S. bovis and the Enterococcus E. faecium and E. faecalis come under this category. So if I go back and revise a little bit, first of all, we had the partial hemolysis. This was alpha. The green color was due to methemoglobin. We had S mutans and S sanguinis, which were part of the viridin streptococca, and we had strep pneumonia, right? I'll show you the alpha one here. So I'll come back to these later on, don't worry. The alpha hemolysis is the S mutants, S mitis, and S pneumonia, the viridins one. Remember, just don't worry about the optotion test right now. We'll come back to it later on. Then we have the beta hemolysis. The beta hemolysis is the complete hemolysis, and this is where you have the agalaxia and pyogenes. Forget the bacitracin sensitivity test and the PVR citrus. We'll come back to it. And then remember the gamma hemolysis. So the gamma hemolysis is where you have no hemolysis, right? No clearing. And the gamma hemolysis basically has your non-Enterococcus S. bovis and Enterococcus E. faecium and E. faecalis, right? Okay. Now let's study the partial hemolysis part again. For the bacteria that are under the partial hemolysis, we need to do a further test to determine whether it is viridin streptococci or S. pneumoniae. Okay, and for that we can do two tests. We can either do the optotion sensitivity or the bile solubility. Now, the optotion is a chemical test that inhibits the growth of S. pneumoniae. So, optotion basically is a chemical and it stops the growth of S. pneumoniae. So think that pneumococcus is basically sensitive, but viridins is resistant, right? So the S. pneumonia is sensitive. It is inhibited by the optotion, right? So how is it performed? You grow the bacteria on the blood agar, you place the optotion disc right in the center, and you incubate. After incubation, if the bacteria stop growing around the disc, there's a zone of inhibition around the disc. Now that means that was sensitive to optotion, which is basically strep pneumonia. It cannot be grown around the optotion. There's a zone of inhibition around that disc. But if it is resistant, then you know it is basically either it is strep mittens or the strep sanguineus or the strep mutans or strep salivarius. Okay, so if bacteria grow right up to the disc, you know it's either viridins or sanguinis or mutants, right? So optotion is a memory trick to remember. Optotion, you can obtain pneumococcus, right? So that's one way of remembering it. I'll paste the memory trick here. I don't know if it helps you, but it does help me. So optotion, whenever something is sensitive to optotion, you know that you obtain pneumococcus, right? Because it's sensitive. So this is the zone of inhibition on an actual plate, and this is your strep mitis, which was not inhibited by the optotion, but the strep pneumonia was. Now, why does optotion basically kill pneumococcus? Because optotion interferes with the ATP production in the pneumococcus species. And the pneumococcus is susceptible to this, but the viridin streptococcus are not susceptible to this. So the S meters still um, survives, right? And then the next test we look at is the bile solubility test. The bile solubility, what it does is this is the second major differentiating test. Bile salts activate a bacterial enzyme called the autolysins. Pneumococcus itself possesses the autolysins, and bile salts activate it, right? They activate these enzymes that the pneumococcus has. So when bile is added, the pneumococcus digests itself, but the viridins does not, right? Um, Now, how is it performed? I'll paste it here basically. Bile salts activate the bacterial enzymes which are autolysins. Pneumococcus possesses these autolysins. When bile is added, the pneumococcus digests itself, but the viridins does not. How is it performed? The bacterial suspension is prepared, and you add the bile salts, and then you observe. If the suspension becomes clear, um, before it was cloudy and now it's clear, this means that the bacteria has dissolved itself. And this means that you have S pneumonia here, and which is bile soluble. But if the suspension was cloudy and it remains cloudy, then you know this is viridin streptococci because it's bile insoluble. So this is another way of saying this. See, this is clear, right? So this is strep pneumonia because it's lysed by the bile. But this is strep mitis. It is not lysed by the bile, right? This is our second test. Now let's actually go to the stuff and try to understand what strep pneumonia is. So what we saw about pneumonia, it was basically lancet-shaped, spear-shaped diplococci. Can we see that here? So it's a bit of a spear-shaped, lancet-shaped diplococci. Lancet is basically a spear. They occur in pairs. You can see they're all occurring in pairs here. It's the first thing to remember about strep pneumonia is that it's encapsulated. Since it's encapsulated, this is the most important virulence factor. Without the capsule, there's no major virulence for this strep pneumonia. With the capsule, it basically evades phagocytosis, it causes the disease, and that's why you need to remember that there's no virulence without the capsule of strep pneumonia. And then you have the IgA protease. Now, where does IgA protease basically live? Remember where pneumococcus lives. So, pneumococcus lives in the nose, it lives in the sinuses, and it lives in the nasopharynx. These areas contain secretory IgA. This organism, Streptococcus pneumonia, this produces IgA protease. So, it basically cuts the IgA. And the result is that the IgA is destroyed, the mucosal defense is lost, and colonization happens, and you get the disease. Right. So diseases are basically, you can remember it by MOPS, MOPS. So M is for meningitis, O is for otitis media, especially in children, P is for pneumonia, and S is for sinusitis. These are the classic pneumococcal infections. Now, important to remember, it produces a rust-colored sputum. Why? Because RBCs leak into the alveoli. Since the RBCs leak into the alveoli, they cause inflammation, capillary damage, blood enters the alveoli, and thus you get the rust sputum. That's why you get the rust sputum, right? Now, why sickle cell patients, basically, why do sickle cell patients get pneumococcal sepsis? Because they become functionally asplenic. Since sickle cell patients functionally become asplenic, the repeated splenic infarctions destroy the spleen. No spleen means capsulated organisms can survive. And the capsulated organisms are especially HIB, uh, meningococcus, and pneumococcus. These are the three capsulated organisms that particularly survive in asplenic patients. Okay, because the capsule is the main part that gives it the virulence. Now, the spleen removes the encapsulated bacteria, but the capsule of this is slippery. Neutrophils struggle to grab it, and the spleen helps this by opsonization and phagocytosis. Without the spleen, capsulated bacteria help and somehow do this bloodstream survival. And this leads to sepsis. Now, at pneumococcus lab findings, we saw that it was gram positive. It was a cocci. It was what else did we see? It was a gram positive. It was a cocci. It was alpha hemolytic. talked about the alpha hemolysis also. We saw it was lancet shaped. We saw there was rust sputum. There were diplococci. They were acting in pairs, and they were bile soluble because the autolysis was happening because of the autolysins. It was optocian sensitive. The growth was inhibited. There was a zone of inhibition around the optocian. That is something you should remember, right? So these are the ways we will diagnose it. Then we have the viridin streptococci. Let's go to the viridin streptococci, right? So the viridin streptococci, again, these were also gram positive. They were alpha hemolytic cocci, but they were optocian and bile insoluble, right? They're part of the normal flora of the oropharynx. These are now you can compare this with the um, strep pneumoniae. Now these are normal flora. The viridin streptococcus are found in the mouth, teeth, and oropharynx. Why is it called viridins? Because viridis means green, and they also produce alpha hemolysis. So that's just one way of remembering that. That's why they're called viridins. Resistant these are. They're not afraid of optocian. Viridins aren't afraid of the shin. That's how you can remember it, if it's helpful. They're bile insolubles. They do not dissolve. Viridins does not dissolve when bile is added. And then you have strep mutans. The Streptococcus mutus is, you can remember it, mutans in the mouth. So Streptococcus mutus lives on the teeth and it produces dextrins. And these are basically sticky polysaccharides. Now, what do the dextrins basically do? The dextrins are sugar, right? So they create this sticky plaque, and that sticky plaque basically becomes dental caries. This is why S mutans is the major cause of dental cavities. Then we have the Streptococcus mitis. Um, okay, so Streptococcus mitis, basically, this is very important. You should remember that the sanguinis makes dextrins that bind to fibrin platelet aggregates on damaged heart valves. Now let's break this down. Now the patient gets a damaged valve. Why will a patient get a damaged valve? That patient either has rheumatic fever or congenital valve disease, or they have a prosthetic valve. There's some way that a patient got a damaged valve in their heart. Now, platelets and fibrin collect here on the damaged valve and they create vegetations here, right? That's how the S sanguinis makes these platelets aggregate on the damaged heart walls. Now, you get some dental procedure or a tooth extraction or cleaning or flossing, and the viridins enters the bloodstream. Once the viridins enters the bloodstream, the dextrins help attachment, right? How does that happen? The viridins enters the bloodstream, dextrin helps that sticky attachment on the valve, on damaged heart valves, right? And the valves get colonized. This causes subacute bacterial endocarditis. It develops slowly over the weeks. And why is it called sanguinis? Because sanguinis means blood. So think blood, heart, endocarditis. That's your sanguinis, right? Causing subacute bacterial endocarditis. That's it for the viridins.